From the Library of Congress in Washington, D.C.
Welcome, everyone. I'm Tomoko Steen at Science, Technology, and Business Division. Today's event is co-sponsored with Office of Health Services. And this is a topic really everyone is concerned about because, outbreak in hospitals, even NIH, and also the food, a lot of antibiotics used in food. So Dr. Price is going to talk about several issues really concerned now. And the UN had a meeting just recently, and that's because, it's going to be out of hand. So it's a worldwide concern. Today's speaker, Dr. Price, is director of Antibiotic Resistant Action Center and professor of George Washington University's Department of Environmental and Occupational Health at the Milken Institute School of Public Health. And he's also the director of Center for Microbiomics and Human Health at the Translational Genomics Research Institute. So this is a nonprofit based in Arizona. And Dr. Price did his PhD at Johns Hopkins University and public health. So I think it's already a little bit delayed. So before further ado, please join me welcoming Professor Price.
Thank you for the invitation and for the introduction. I appreciate that. I'm not checking my email. I want to make sure I don't go too far over time. So I think you recognize that this is the world we live in today, right? The world between the freak out moments. So this could be applied to, perhaps gun control and these horrible school shootings where, these take place, we freak out, we demand action, and then we go back to our lives until it happens again. This could also be applied to weird weather events and global climate change. And some have argued that this applies to superbugs. So we do hear about superbugs. They hit the mainstream media, and we freak out for a little while, and then we go back to our busy lives, and we expect that somebody did something about it, right? But I would argue that this is actually the graph that applies to superbugs, because what happens is that these things hit the mainstream media, they're circulating, we're hearing about them, and we expect the CDC or somebody to do something about it, and then it kind of leaves the media cycle, right? And we think that they've gone away, but in fact they're still there, and when the next one comes out, it's piled on top, right? And what's happening is that the medical community is just learning how to deal with it. I'm going to learn how to deal with this. Okay, stop. Thank you. All right, so so the medical communities, doctors learn, they scramble, they reach higher on those shelves of antibiotics, and they reach for the next one and the next one, and they get the stool out and they reach out for the next one. But what's happened is that we are running out of shelves, so we don't have new antibiotics, and the bacteria are continuing to evolve resistance. And so we keep hearing now about us moving towards what's called the post-antibiotic era in medicine. And the World Health Organization describes this as a time when even a scraped knee can result in a deadly infection, because you can get Staph aureus in that scrape, and if we don't have antibiotics to treat the Staph aureus infection, that can get into your blood and kill you. A bladder infection that's really a painful annoyance right now can progress to kidney infections, and once it's in your kidneys, it can access the blood and cause deadly blood infections. And so we're moving towards a time when these really common infections can be deadly. And I think, so this is not the apocalypse, right? So it's not the end of humanity as we know it, but I don't think it's overstating it to say that this is going to change our lives dramatically. It's going to change everything from what procedures can take place in the hospital. Can we do that hip surgery? Can we do that bowel surgery? Can we give somebody chemotherapy that's going to knock out their immune system for several months? Probably not. And it's also going to change the way it feels to ride public transportation or ride that escalator and grab that handle and think about whether you picked up a superbug or not. Right? So these superbugs have many different monikers, right? You hear about MRSA. Who's heard about MRSA? MRSA, right? Methicillin-resistant Staph aureus. Kills 11,000 people in the United States each year. VRSA is just vancomycin-resistant MRSA, right? And vancomycin is a crappy drug, but this is the one we're really afraid of because that's the last drug we've got. VRE, CRE, these are just different monikers for superbugs. And these are just basically disease-causing bacteria that are resistant to antibiotics, usually multiple antibiotics, although we usually refer to them based on one drug. And so people ask me all the time, so how does this bacterium, that's a bacterium to me, how does a bacterium become a superbacterium? That's a superbacterium to me. How does it go from this to this? And it goes from this to this by changes in the DNA. So bacteria multiply themselves. So they don't reproduce sexually. They just make copies of their DNA, and then they split from 1 to 2. And then 2 become 4, 8, 16, 32. And each time they do that, they have to make copies of 4.5, sometimes more millions of bases. So 2 to 4 to 5 million bases of DNA, G's, A's, T's, and C's. And when they do that, they can make errors, and some of those errors result in changes in proteins that make them resistant to antibiotics. So the other thing that they can do is some bacteria carry around these extra little packages of DNA called plasmids or other elements, and they can pass these to other bacteria. So in a single genetic event, they can pass resistance to 6, 7 different antibiotics just by bumping together and passing this DNA. And now what we have is we have a world where we have bacteria that are already carrying lots of different resistance genes. And they're just waiting for that one last resistance gene to get that royal flush, right? And this is what we're really afraid of, right? We have lots of bacteria that are one card away from a royal flush. And now we know that that last card is floating around out there. I'm going to come back to this later. So that's happening all the time in the background. But most of the time, that's a dead end, right? So these mutations happen, and then those bacteria fizzle out. But if you're in an environment where there's lots of antibiotics, even one of those mutations can result in a rapid evolution of drug-resistant bacteria. So in an environment where you have lots of drugs, then the susceptible bacteria die off. Excuse my sort of South Park-style cartoons, but these are the best I can draw. And that resistant one then goes on to multiply. So 1 becomes 2. I skipped a step, 2 becomes 4, 4 becomes 8, etc. And you can go from a single cell to a billion drug-resistant bacteria in 24 hours. So E. coli can do these doubling every 30 minutes. And E. coli is one of the worst superbugs that we deal with today. So you can go from a single cell, superbug cell, to billions in 24 hours. So there's a lot of confusion in the mainstream media. You all look brilliant, so I probably don't need to say this. But the media talks about people becoming resistant to antibiotics, but I think it's really important to make sure that you understand this, even though it's a simple concept, that it's not people that are becoming resistant to antibiotics, it's bacteria, and those are colonizing us and infecting us, and those infections are harder to treat. It gives doctors fewer choices, and these result in more and more deaths every year. And the CDC conservatively estimates today that 23,000 Americans die each year of superbugs. And Tom Frieden, the head of the CDC, underscored several times that this was a conservative estimate. It's really difficult to know whether somebody died with a drug-resistant infection or of a drug-resistant infection when somebody's already sick. So, understanding that rapid evolution, understanding this relationship, underscores the fact that each time we use antibiotics, we potentially fuel the growth of superbugs. And Stuart Levy, one of my heroes from Tufts University, coined this term. He said antibiotics are societal drugs. So what he was trying to do was explain that antibiotics are different from any other drugs that we use. And I like to start with a counterexample. So Tylenol is not a societal drug. People take Tylenol for headaches. If you misuse Tylenol, if you overuse it, if you use it while you're drinking, you can get liver damage and die. People commit suicide, a horrible suicide, using lots of Tylenol. But it doesn't affect anybody else's ability to take Tylenol for a headache. However, if you misuse antibiotics, you can drive the bacteria in your system, in your gut, on your skin, to become resistant to antibiotics, and you can share those bacteria with other people, preventing them from using those same drugs. So that's what Stewart was trying to convey with this idea of the societal drug. Now, when you think about that societal nature, you have to recognize that the context and the quantity of antibiotics that we're using really matters. And I think the thing that people think of most of the time is this context, right? You think about the hospital, and this is where we know there are lots of drug-resistant bacteria. And we know that if these people are not washing their hands and aren't hitting that hand sanitizer between every patient, they can become vehicles for drug-resistant bacteria. They can spread them around. And we have to have teams of people wiping surfaces all the time to keep those contact surfaces clean. So nobody denies that. And nobody denies that this is too much. We're using 8 million pounds of active ingredient. That's not the size of the pill, right? That's the active drug inside the pill. 8 million pounds of that in the United States in human medicine. American Medical Association, American Academy of Pediatrics, all these different associations say this is too much. We need to cut it back. We're overusing antibiotics. And why do they say that? Because we're fueling the growth of superbugs. And then if you have the combination of this excessive use and then poor hygiene, the doc skips that hand sanitizer, skips washing the hands, then that's how we have these horrible hospital outbreaks of drug-resistant bacteria. So that's human medicine. But I want to talk about animal production. So that's the thing that I spend most of my time studying. And here's the quantity in animal production in the United States. This is the most recent number, and we were hoping it was going to go down. It went up. So we're at 34 million pounds of antibiotics in food animal production in the United States each year. So you can see that that's more than what we're using in human medicine. So let's talk about the context. So that's quantity. So this is not the context. This is my imagined context when I was a kid. And this was, anybody play with these when you were a kid? Yeah. All right. So you can buy these on eBay. It turns out I found the era that I played with and I took that photo. Okay, but this is the way we actually raise animals today. So cattle will spend part of their lives grazing in open pastures, like my family's ranch in Central Texas, but they'll end up on feedlots like this one. And that's not dirt that they're standing around on. Pigs spend their entire lives in concentrated animal feeding operations today. And these are not full-grown pigs. When they're full-grown, they are snout to tail. But they're so stressed out in this environment that they clip the tails off because they'll bite them off. They get very aggressive under these conditions. Chickens, up to 100,000 in a single house, and turkeys are raised like this. So some people refer to these as factory farms, but as a microbiologist, I don't see factories making meat. I see factories making lots and lots of bacteria, right? And when you add that magic ingredient of low levels of antibiotics, then you have a factory making superbugs. And this has been established with every study that we do around these factory farms, around these industrial farms. We detect lots and lots of drug-resistant bacteria. Now, one of the things that we use the antibiotics for, and one of the major things that we have historically used antibiotics for, was to make animals grow faster. So we give them low doses of antibiotics. This was discovered in the 1940s, like right after Alexander Fleming discovered antibiotics. Somebody discovered that giving animals low doses of antibiotics helps them grow faster. We don't really understand, probably changing the microbiome, that is the gut community of bacteria, passing more energy on to the animal. But we don't exactly know how well it works and how it works. But the other justified use is preventing diseases. But some of the diseases that we're preventing are what we call production diseases. That is, diseases that are occurring because of the way we're raising these animals. So I'll give you a couple of examples. So one of the things that we have a lot of is shipping fever in cattle. So it turns out that when you raise animals, you let the animals graze, you have to then get them to those feedlots, and you put them in these trucks to get them to those feedlots, and sometimes you're shipping them for days. And you stack them up two layers high. These are cattle actually in this. This is what it looks like when you empty it. And so it doesn't take a microbiologist or an infectious disease doctor to recognize that this is going to make these animals sick. But rather than change this system, don't ask me how, maybe distributing smaller feedlots closer to the grazing lands. Instead of changing that, we just give them all antibiotics, right at a time when we're about to put them all together in those fecal wastelands. So that's one production disease that we're treating. Another one is when you switch these animals that have evolved to eat grass and you switch them to corn and soybean, they get liver abscesses. And so rather than trying to figure out a way to have them not get liver abscesses by changing their diet or lengthening the time that we're feeding them, we give them antibiotics, again, while they're crowded together. Okay, so here's a problem. I think in Fisher-Price, maybe that's not a problem, but the public health problem that I want to describe is that each animal has trillions of bacteria living in and on it. So that's not trillions, but I don't have enough pixels to draw this. But anyway, the animal has trillions of bacteria living in and on it, and when you feed them antibiotics, you're going to force that evolutionary process to take place in those animals. And so the bacteria become resistant, and then when you chop up those animals to make pork chops, you're going to inevitably contaminate some of those pork chops with some of the bacteria from either the gut of that animal or the skin of those animals. And then those bacteria can go on to cause infections in people, and we get the little frowny faces. And again, the problems that I described before. You're giving physicians fewer choices for treating those infections, and we know that those infections last longer, and they cost more to treat, and they result in more deaths. So I just want to underscore something because, again, there's been a lot of confusion about this issue. The main concern, while we are concerned about traces of drugs in the meat, it's not the antibiotics in the meat that we're most concerned about. It's drug-resistant, living, infectious, drug-resistant bacteria on the meat. That's the real public health challenge. And so sometimes you'll hear companies describing how they remove antibiotics early enough so there's no drugs in the meat. So don't worry. But that doesn't, the bacteria don't respond that quickly to that removal. And so when you go to slaughter those animals, you're going to contaminate those bacteria, or the meat with those bacteria. Okay, so for years it turns out there's been a big argument about this for years, and I've been sort of part of this argument. Not as long as people like Stuart Levy. Stuart Levy's been having this argument for 40 years. But for years, the argument was framed around the classic foodborne pathogens like Salmonella and Campylobacter. I'm sure you've all heard of Salmonella. Have you heard of Campylobacter? Okay, it turns out that they are sort of tied number one and number two in terms of causing diarrhea in the United States, bacterial diarrhea. And together they cause about 2.6 million cases of diarrhea, all right? And most of that is coming from contaminated poultry products. And the CDC estimates that about 410,000 of these infections are antibiotic resistant. And that resistance can be traced back to either contemporary or historic use of antibiotics in food animal production. So just to put things in perspective, that's the population of Miami getting drug-resistant foodborne infections in the United States each year. But not very many people die of those infections. So most people who get diarrhea will have, they'll be uncomfortable for 5 or 6 days, they'll use a lot of toilet paper and they'll recover. 1,000 people will not. They die. It gets into the blood and they die. But apparently 1,000 people is not enough to change policy in the United States. So we have to move. I've been saying for years that we have to move back. We need to move beyond the classic foodborne pathogens because there's a lot more than Salmonella and Campylobacter in the food supply. We need to understand what we call the colonizing opportunistic pathogens, or COPs. These are the big killers. And when you talk to an infectious disease doc, they want to know about these guys. They don't care about Salmonella and Campylobacter. They just don't kill enough people. So we're not talking about Andy Griffith here. We're talking more along the lines of Harvey Keitel and The Bad Lieutenant. Some of these movie posters really give it all away, right? Gambler, thief, junkie, killer, cop. All right. Anyway, it's colonizing opportunistic pathogens. These include things like E. coli, extraintestinal pathogenic E. coli, the kind that causes bladder infections, Klebsiella pneumoniae. Have you heard of Klebsiella? A few of you. So this is a big killer in the United States. One of the biggest superbugs is Klebsiella. One of the biggest superbugs is also an E. coli. And one of the biggest superbugs is Staph aureus, MRSA, MRSA. So COPs can live in or on our body. So staph can live in your nose, in your throat for years without you knowing it. No symptoms. You can just be a carrier and you can share it with other people. E. coli can live in your gut for decades and you wouldn't even know it. It doesn't cause symptoms until it gets into the bladder, until it gets into the blood. And so you can become this reservoir. And so these COPs cause what I like to call insidious epidemics. So they can spread globally. They get on planes with us. They get on trains and boats, and they get on meat, and they travel the world. The problem is that they have asymptomatic colonization. So these fever monitors in airports are not going to work, right? They're not going to detect these things. There's silent person-to-person transmission. You come home from your trip to India or China or the United States, and you bring home drug-resistant bacteria and share them with your family members. And no one knows it, right, until their immune system's knocked down. We don't know how long they can colonize. We know that they can be from days to years. And then it's really difficult because you can have animals carrying them, you can have humans carrying them, you can have environment, water carrying these bacteria. So these are really hard to study, and I like a good challenge, and so that's what I study. And this is the machine that I use. This is a next generation DNA sequencer. So now we have these sequencers. We can sequence DNA at a rate, at a cost that was never even imaginable before. We can sequence a bacterial genome, millions of bases, for less than $50 today. When the anthrax letters were mailed and we did this investigation on the anthrax letters, it cost $500,000 per genome, $500,000 per bacterium. Today it's $50. This is changing fast, and it's changing what we understand about infections, and it's so fun. I have to tell you, I'm a geek. Okay, so what we do is we take the DNA, and then looking at these little mutations, we can redraw the family history of these bacteria. So if we have bacteria, so going back to the anthrax letters, if we have a letter and we have a vial or a flask in a government lab, and we want to know if these two are related, we can sequence the DNA and we can look for these little mutations and we can see how they're related to one another. So we draw these little trees, and we just have, and based on the length of these branches and what we call these common ancestors, the little nodes connecting these guys, we can redraw the evolutionary history of these bacteria, and we can understand how they're related to one another. And then we can actually do something that's called rooting, and we can understand who is older, or which nodes are older and which ones are more derived, that is, more recent. And this is so powerful. It's almost like a time machine, so we can go back in time. And we need a time machine for this because we've been using antibiotics in animal production for 70 years. So how much of the drug resistance that we're dealing with today in human medicine is due in part because of antibiotic use in animal production? These time machines are helping us figure this out. So I'll tell you a little bit about the work that we're doing on MRSA first, and then I'll go on to E. coli. So MRSA, this is, microbiologists like me are weird, so they're stuffed animals. Have you seen these giant microbes? Yeah. Okay, so there's these giant microbes, teddy bears that you would hug. I don't have one. But if you want to learn about MRSA, I really recommend Maren McKenna's book, Superbug. It's a very long title if you read it, but I just focus on the green part, Superbug. It's a really, really well-written book. It reads like a novel, but it's a nonfiction. But if you don't have time for that, you can just go to the CDC website, and you can learn about the 5 C's of MRSA transmission. These are the things that we know lead to the transmission of MRSA. So crowding, frequent skin-to-skin contact, so rubbing up against one another, compromised skin, so cuts and abrasions, contaminated items and surfaces, and then a general lack of cleanliness. The last two seem a little redundant to me, but I'm not criticizing the CDC. Okay, they go on to say where the 5 C's are really, really prevalent, and I guess you would want to stay away from these places. So the 5 C's are common, and locations where the 5 C's are common include schools, dormitories, military barracks, households, stay away from those, correctional facilities, and daycare centers, right? So all the places we kind of hang out, right? And I think what they're getting at is that where you have crowding, where you have people living close to one another, you can pass these bacteria along. But when I see the 5 C's, I don't think about households. I think about a concentrated animal feeding operation. Now you have full-grown pigs. This is the way they look when they're full grown, just about ready for slaughter. So this is what we call a concentrated animal feeding operation: crowding, contact, compromised skin. They bite each other. They get abrasion from the fencing. Contaminated items and surfaces. I'd say pig poop, by definition, is sort of, a lack of cleanliness. So it's no wonder that we've seen the emergence, just in the past decade or more, of something called livestock-associated MRSA. So new strains of MRSA directly traceable back to livestock production. We call these LA MRSA. So these disproportionately affect farmers, farm workers, veterinarians, and actually the people who live with those people as well. So a few years ago, we decided we wanted to understand the origins of pig MRSA. So we collected MRSA strains. This is MRSA ST3. This is sequence type 398. This is just a geeky label that people like me put on bacteria. I'll talk more about sequence types later. But we collected all these, and we collected them from people and from animals, and those that were MRSA and those that were susceptible to methicillin. And we sequenced the genomes of nearly 100 of these, and then we drew an evolutionary tree to understand how they were related to one another. And so this was the tree. I don't expect you guys to really memorize this, but each of these nodes, each of these little names, the little digits at the end, is a different isolate, that is a different staph bacterium. And then pink is from pink is from animals, and gray is from people. And you see that the pink are all clustered up there. It turns out that that's the livestock lineage, and these down here are the older ones. Those are the livestock independent lineage. Those are the ones in people. And so what we learned from doing this was that the livestock-associated MRSA actually evolved from staph and people. So I have a cartoon version. I think this is better, right? So Fisher-Price is better, even for Nobel laureates, they've told me. All right, so it turns out we have staph circulating in people. Those people inadvertently pass staph to the animals. So, this is kind of gross, but, farmer blow? Snot rockets. Anyway, you can share bacteria with your animals from your nose, possibly. And then where we're using lots of antibiotics in animals, we get this transition from MSSA, so susceptible, to MRSA, MRSA. And then that's spreading now back to people. And this is a very clear, we have this epidemiology is very clear now that this is happening. What's also happening, and I mentioned this before, is that the people living with these people are at very high risk for picking these up as well. But the story continues to evolve. So we're studying this very intensely in Northern Europe, where we have access to the animals, to drug records, to everything. Whereas in the United States, we get a shotgun in our face when we try to go onto these farms. But there we can study these very clearly. And we see now it's spilling over into the general community. It's usually the people living near these farm workers or around the farm. So it could be environmental exposures, or it could be secondary exposure to these people. We've seen outbreaks in hospitals that are clearly livestock strains. And it's from these people going into the hospital, and then a mistake with the hand hygiene probably, and they pass it on to other patients. And then now we see for the first time, we just published this 2 weeks ago in the Clinical Infectious Diseases, a really important journal in my field, that there's evidence of foodborne transmission too. And this is the first time that we've seen this. So this is continuing to evolve. We see that the DNA of these bacteria are changing, and they appear to be becoming, some strains are becoming more adapted to humans, and so they could spread better among humans. And so we're keeping our eye on this, and we're using these DNA technologies to keep watching it. It's now become a textbook example, though. So this is my little cue to remind me to say this. This is a textbook example of what happens when you use antibiotics in livestock production, what can happen to people. And so when I spoke at the CDC back in January, and I was just, going over my slides, and I put this slide in, and I said, I should just double check that they still have that up on their website. And I went there, and they'd modified it, and they added this. So they added, and areas where animal handling is common, such as veterinary clinics and livestock settings. So they saw our paper, I guess, and many other papers coming out on livestock-associated MRSA, and they modified their website. The CDC these are real scientists, and they are an amazing group of people really looking out for our health, and I thought this was very cool. Okay, I'm switching to E. coli now. So staph is the one that lives on our skin and our noses. E. coli lives in your gut. And E. coli has a lot of personalities, right? There's thousands of strains of E. coli, but from a clinical perspective, there's three. There's the good, the bad, and the ugly. So the good is the kind that about 30% of us carry in our gut now. It's just part of our microbiome, probably just helping us with digestion. The bad is the kind that, like the Jack-in-a-Box E. coli from the early 90s, right? So these dramatic foodborne outbreaks, Jack-in-a-Box responded very well and helped have something called HACCP, which is Hazard Analysis and Control Point. So this really revolutionized food safety in the United States. But we still have these outbreaks of the bad E. coli, the diarrhea-causing E. coli, and about 50 to 100 people die of that E. coli each year in the United States. But the ugly, the really, really bad one, or super bad, as I like to say sometimes, is the kind that causes urinary tract infections. And this one kills about 36,000 to 40,000 people each year in the United States. So let me tell you a little bit about XPEC, extraintestinal pathogenic E. coli. It has special features. It has attachment factors. It has these little hooks on its surfaces to attach to urethral cells. It can cloak itself from the immune system, so it can hide itself from the immune cells that want to gobble it up. It can produce toxins. It can acquire iron. It can survive. It can deal with uric acid, so it can deal with the harsh environment of the bladder. And it causes 6 to 8 million bladder infections in the United States each year, 250,000 kidney infections at least. I think this is an underestimate, and these numbers are getting old. This is 2006 numbers. And also deadly blood infections. So as I said before, about 36,000 to 40,000 deaths. In 2006, the numbers almost certainly higher today. And antibiotics are critical to prevent this ascent from the bladder into the kidneys and then into the blood where it kills you. And so antibiotic resistance is a major challenge when it comes to XPEC. So I want to lock in this relationship because not many people, I didn't know this until I met this guy named Jim Johnson, a wonderful professor at the University of Minnesota. And he's like, Lance, you need to be looking at E. coli because E. coli causes all these bladder infections. It turns out it causes 80 to 85% of bladder infections. So E. coli is the main uropathogen. The gut is the immediate reservoir, so it's really E. coli getting from the gut into the urinary tract. I'd love to talk for a while about how that happens, but I mean, there's lots of ways that happens. Sex is one way, the best way, I think. So it's much better than riding a bike in dirty underwear, I think. And then, so understanding that it's the bacteria in the gut getting into the urinary tract, and women, I won't ask for a show of hands, but women are at much greater risk for urinary tract infections, at least until we get into our 70s. So understanding how these things then get into our gut is really important. And I, along with a number of people, real pioneers in this field, have been asking this question: is it possible that foodborne bacteria, E. coli, are causing urinary tract infections? So this guy, Lee Riley, in the middle, was the first one working with Amy Mangus on the end there to ask this question. They recognized these outbreaks of urinary tract infections, and they said, this looks like a foodborne outbreak. But we're still been dancing around this a little bit, and it's really the genomic methods, and I want to tell you a little bit about what we're doing to really resolve this issue. That's Jim Johnson between Amy and Lee, and this is Niels Fridmont Moller from Denmark, who also does really wonderful work on this. And they're all friends and collaborators. So in 2012, actually 2011, I had this idea that I want to do this study to understand this, to quantify this, and to prove whether it does or does not happen. And so I was living in Flagstaff. That's where my lab, one of my labs is in Arizona, and it's a small, geographically isolated city. And we said, let's go to every grocery store in the town, twice per month and buy every brand of chicken, turkey, and pork. And let's team up at the hospital where my friend, my old classmate, runs the infectious program or the infectious disease program. And let's collect every E. coli from every urinary tract infection and blood infection over the course of a year, the same year, in the same town. And let's see if we can resolve how much overlap there is. And so we did that, and at the end of the year, we'd sampled nearly 2,500 meat products, and we'd got almost 1,300 urinary tract infections from Flagstaff Medical Center. So this is what we found in the meat supply. It turns out most of the meat that we tested, 81% on average, was contaminated with E. coli. No surprise, E. coli is an indicator of fecal contamination. You have feces in your meat. Sorry. Sorry, cook it, wash your hands. But nearly 90% of the poultry products that we tested were contaminated with E. coli and 60% of pork. All right, we looked at then, what were these resistant to? They were resistant to lots of different antibiotics. And so the blue line, the blue column is from chicken. So these are the E. coli from chicken. Each of these 3-letter codes is a different antibiotic, and the percent is the percent of the E. coli that we tested that was resistant to these different antibiotics. And what you see, you will notice that turkey is off scale, right? Why is that? Well, I would guess that it's because they're using more antibiotics than these other commodity producers, but in the United States, we don't have a right to know what antibiotics are being used in which animals because that's a trade secret. Antibiotic use in animal production is a trade secret. It's their secret for making cheap meat for you guys. Now, don't have that sour look. The one place where gentamicin is, the one place where chicken beats turkey is actually gentamicin. And that's because it's legal to inject chicken eggs with gentamicin in the United States to prevent infections from possibly occurring. So we have these machines. We have machines that inject pallets full of eggs simultaneously, while we're also administering vaccines. Vaccines I love. I think that's a good idea. Injecting antibiotics into eggs, terrible idea. And we did a study of people who catch chickens for a living, and they had 32 times the risk of carrying gentamicin-resistant E. coli as their community peers. So now we're asking, are these bacteria related? So we have 1,900 or nearly 2,000 E. coli from the meat. So 81% of all those products that we tested, we got E. coli from there. And then we have E. coli from urine and blood in the hospital. And now what are we going to do? We're going to sequence all those genomes. I told you it got really cheap, right? And we had a $1 million grant. We spent it in 2 years. We had a $1 million grant to do this. And so we sequenced all of those genomes using one of these next generation sequencers. So we have a 3-step approach, and this is kind of wonky, so just roll with me for a minute. I'll boil it back down with Fisher-Price, or at least I'll try to be simple about it. So what we do is we sequence the genomes, and then we do a low resolution genotype, so using a standard method called multi-locus sequence typing. It doesn't matter, but we assign them to a sequence type so we can group them together, and then we use the whole genome to make one of those family trees. If we try to do them all together, we lose resolution, actually. So this is the way that we do it. We group them, then we make a family tree. We group them, we make a family tree. And we ask at a very high resolution, with the best resolution you could ever hope to have using the whole genome sequence, how are these related to one another? What we see in the food and what we see in the people in the same city over the same course of the year. Okay, so here's the first thing. So this is just that initial grouping and how they're related to one another. This is really cool. So the size of the bubble is, the number is the sequence type. So that's a DNA fingerprint. It doesn't matter. It's like, consider it like a name, Fred, Jane, Larry, whatever. And then the size of the bubble means that there were more of the E. coli with that name. So sequence type 131 is one of the most important E. coli causing urinary tract infections in the United States today, actually globally. And we did a study where we showed it's a brand new strain that's gone pandemic in a very short time. And then if it's yellow, it's urine. If it's red, it's meat. That's not by accident, right? So meat is red, yellow is urine. And then if they have a little line connecting them, they're related to one another. And I think what you can see is that you've got your reds all clumping together, and you've got your golds all clumping together. So these are all related together to one another at some level. But what you see is you have Pac-Man shaped bubbles, right? So you see these red pie pieces in the gold bubbles, right? The yellow, urine yellow bubbles. And you see yellow pie pieces in the meat red bubbles, right? Suggesting population overlap there. But at this level, this is low resolution, so we can't say with confidence that, yes, meat was the source of that infection from 117 or those 131 infections. We can't say that meat was definitely a source. But what we can do is we can take these little bubbles and we can draw that high-resolution tree, and we can actually look at it in great detail. I hope you guys can see the color. Can you see that there's red and black? So black is from a urinary tract infection. Red is from meat. So gold won't show up, right? So I had to stick with black with this one. All right, what you see is you have two different lineages. They're distinct, right? So we would call none of these urinary tract infections, we're not going to call any of these foodborne. We're using a very conservative view of this, and we're saying these are two separate lineages. They are unrelated. So let me show you another one. There's ST58, so that's the name of that one. Let's draw a tree with that one. You see that the urinary tract infections, the black ones, are nested all throughout the red. They all share common ancestors with the food. And those are very short branches. That means that they all came from the same population very recently. And so these are foodborne urinary tract infections. So we do that with each of these bubbles. We go through each one. ST95 looks promising, right? It looks like there could be some overlap there. But it turns out that, and I know you can't, I realize now that you can't see the color, but the only red you see is up there all by itself. And so we're going to conservatively say that these are unrelated. It's possible that they are related, but we're going to be very conservative for this first round of estimating how many urinary tract infections are coming from food. Because I can tell you there's an industry that does not want any of these to be foodborne. So very conservatively, the blue circles, and those were supposed to be hidden until right now, it was a surprise, but it didn't work. The blue circles represent all of those bubbles where we have very clear, very close, high-resolution relationships. So each of those represents foodborne transmission, in our opinion, my opinion, and my colleagues. So today, now these numbers are not final. If you were the media, I wouldn't show you these data yet. But this is our most conservative estimate, that 9% of the urinary tract infections in Flagstaff, Arizona, and Flagstaff, Arizona is very representative of the rest of the country because all the meat that we produce is produced in these local areas and then distributed throughout the country. So 9% is our very conservative estimate, and it may be as high as 12 plus. When we loosen up when we loosen it up, it's 12 plus. So we're talking about now, if you scale that to the rest of the country, we're talking about over 500,000 infections at the most conservative estimate, 500,000 to 700,000 infections that we had no idea were foodborne. So this represents a totally different paradigm and a new conduit for resistance genes, from a newly recognized conduit of resistance genes from animals to people. And I can tell you the other thing that we haven't even looked at is the resistance genes moving independently, which is another thing we have to look at. So I just want to stop here for a second, and I know I always see confusion when I talk about foodborne urinary tract infections. So what happens is that you mishandle the poultry, or you consume undercooked poultry or some other meat, and you consume the E. coli, it gets in your gut, and it grows there for a little while, and then it makes the short trip from, sorry to be graphic, from the anus to the urethra, and you have a bladder infection from food. Okay, does that make sense? So we've coined this term, foodborne urinary tract infections. I like to call them foodies. Foodies against foodies. And this is truly a paradigm shift in what is a foodborne or food-associated infection, right? It's no longer, sorry, I'm a microbiologist. It's no longer diarrhea and vomiting, right? Now it can be a MRSA infection. It can be an untreatable bladder infection. And it gives us new context for those 34 million pounds of antibiotics that we use in the United States each year. But we are not alone, right? And we've distributed this model. We've shown this model of industrial animal production to the world. And the world is changing their consumption habits. So the BRICS nations, Brazil, Russia, India, China, South Africa, these countries are developing very quickly, and they're adopting our meat-centric diet, and they're adopting our methods of producing animals in these industrialized systems using lots of different antibiotics. And this is a global challenge that we all have to meet. And the problem is that we don't have a box to stand on because we've been doing it. We designed this system, and we really haven't changed much over the years. And my birthday's November 19th. I don't know if you can see this date, but November 19th, this hit the media cycle. So a new mobile resistance element was found in animals and people. So it turns out that China had adopted our methods of using antibiotics in food animal production. They were using our last drug, a drug called colistin, our last drug for treating CRE, carbapenem-resistant Enterobacteriaceae. That's super E. coli, super resistant E. coli, and its cousins, Klebsiella and those guys. They were using that drug to raise animals using our model because it was an old drug. Nobody cared about it until we ran out of drugs and we got desperate and we started using colistin. And then all of a sudden we realized that this new gene had evolved. We'd never seen it before. It was on a mobile piece of DNA, a piece of DNA that's very readily transmittable between different strains of E. coli and Klebsiella. And so this hit my field like a ton of bricks. And in the weeks that followed, we had my colleagues around the world started scanning their databases of DNA, and they said, hey, it's here already. Hey, it's over here already. The highest concentration that we've seen is in China, where they were doing this. But we found it now. It's already in the United States as well. And this is the last card for that royal flush. So for CRE, some strains of CRE are just missing one gene, and this is it, to be untreatable. And so now we're talking about untreatable bacteria on meat, but we don't even recognize the kind of E. coli, this kind of E. coli as foodborne pathogens. So how do we regulate that? How do we say we don't want your product? And how do we respond to China and other countries that are using our last drugs for food animal production, or any drugs, in an abusive fashion? How do we respond to them when we do the same thing in the United States? It's really difficult. So I'm just wrapping up. I just have a couple of final thoughts, and then we can talk. The flyer said antibiotics good or bad. Antibiotics good. I mean, antibiotics have saved countless lives since their discovery, since Alexander Fleming accidentally discovered penicillin in the 1930s, and they were introduced to human medicine. Yes, they have a profound effect on our microbiome. That is the community of bacteria that we actually are, dependent on. But our microbiomes generally recover from those temporary things, and these antibiotics save us from the acute infection that we're suffering from. Someday we might have something better that specifically kills just the bad bacteria and leaves the good bacteria intact, and those are some of the things that we're trying to work on at GW. But for now, this is the best thing that we have, and we should really protect them. And then the other thing I want to say is that sick animals need antibiotics. So I don't want anybody to walk out of here thinking that I'm saying that we shouldn't treat sick animals with antibiotics. We should. But if we have a system that makes animals sick by design, that system's broken, and we have to change that system. And we may not have to go to something as radical as free-ranging, happy little pigs, but we should be raising them in a way that doesn't make them sick repeatedly. I mean, if you can predict it based on a clock or a calendar that they're going to get sick and you have to add antibiotics, that system's broken, in my opinion. And the last thing I want to end with is a message of hope. I don't think that the post-antibiotic era is inevitable. I mean, some people have already reached it. 23,000 Americans die each year, right? But this time when there's just no antibiotics that work, it's not inevitable. I just came back from Scandinavia. They've really control antibiotics in animals, they really control antibiotics in human medicine, and they have really good hygiene in their hospitals, and they have really low drug resistance rates. And the people who have drug-resistant infections are often returning from travel or have consumed imported meat products. So there is hope, but we have to do this globally, and I do think we can do it. So just, none of you guys care about this, but this is, science takes massive teamwork. I have computer scientists, I have clinical research coordinators, I have clinicians, I have lab technicians, I have very dedicated people that work with me, and I appreciate them all, so I can just go out and talk about it and draw Fisher-Price characters. So with that, I'll stop. Thank you. Yeah, I'm very happy to take questions. I'll stay as long as you want.
Could you repeat the question also?
Mm-hmm.
Any questions?
How do antibiotics kill bacteria?
So how do antibiotics kill bacteria? There's lots of different molecular mechanisms, but on a very high level, they either block DNA synthesis, RNA synthesis, so that's DNA goes to RNA, goes to proteins, they block protein synthesis, or they block the cell walls that the bacteria need to hold themselves together. So they're blocking the essential steps of making a bacterium. Yeah.
Other question?
So it's kind of a practical question that when we go to hospitals and when doctors prescribe us antibiotics, they give the suggestion of dose, frequency, and the length of time that they take it, and what reference they have to give those suggestions to patients.
So your question is one of the things that doctors say is that you should take an antibiotic for a specific period and only for that period?
And research based or it's continually improving?
Yeah, so I think that's something that they're really trying to dial in. I mean, it's not a perfect science yet. Like, how long do you take them, and how much do you take, right? So it's rarely adjusted for body mass size, and you've noticed in the United States that body masses are changing, right? But what we're learning is that, some drugs we're learning that you can cut back the amount of time that people are on, so that you reduce that pressure on all the bacteria in the person, right? So you can dial that back, but you should take them for as long as the doctor says you should take them, and let them figure out what that time should be. But right now, if they say 10 days, take it for 10 days. If they say 3 days, take it for 3 days. Did I answer your question?
Right, I just think maybe it's not optimized in regards to being treated ended up unnecessarily, so that the overdose of antibiotic actually help to, spawn this resistance among population.
Right. So I agree. Both dosing and who gets antibiotics is not optimized, right? So doctors often prescribe what we call empirically, right? So somebody comes in and they have some symptoms that could be or could not be bacterial infections, but they don't have time to collect a culture and wait for the culture, or they don't think they have time to do this, collect a culture, wait for the culture to come back, check those records, and then make a reactive recommendation. And so they just guess, and they say, you probably have E. coli in your bladder, and it's probably susceptible to this. Here's a prescription, get out of here. And so what we really want to do is move towards a rapid molecular diagnostics, so they can collect a pee in the office, and within 15 minutes know exactly what you're infected with, what it's susceptible to, and give you the exact antibiotic that you should have for the exact duration. And I think we'll be there in a few years, actually. Yeah, I mean, the molecular techniques are out there. Yeah. You had a question back there. Yeah.
Well, I know you're saying a lot of it is family. So what is your recommendation for maybe, what's your recommendation for maybe staying clear of that induced?
What should you do? Is that what you?
Should we go organic? Should we give up meat? And what is your recommendation since it is being found a lot in food?
So I'd be a hypocrite to say that you should give up meat, although it is a healthy choice. I'm not a vegan or a vegetarian, but that is one way to avoid foodborne drug-resistant bacteria. The other part of this, though, is that 9 billion animals make a lot of poop, and that poop is used as fertilizer on crops where they grow vegetables that vegetarians eat, right? So this is an ecological problem. So we don't, consumer choice helps, but we really, we need to tackle this as a global community. But yeah, so what I do is, personally, is I only buy products that are from animals raised without antibiotics. So I look for that label. That's the only label that really matters there. Organic is also a label that indicates that they have not been treated with antibiotics. And then there's a new label that's coming out that's responsibly used. What's the actual term? Anyway, there's a responsible use term that they're working on now, and that has to be, we're really pushing for that to be only third-party verified. So it has to be the USDA verifying that they're actually using them responsibly, which would only be to treat sick animals. And so we need, I'm really leery of this, but I do think that we need to get there eventually, is allowing for the treatment of sick animals and still selling that, with a specific label. But it has to be third-party verified. We can't count on companies to make that designation.
Right, because I was told with organic there's certain levels of organic, so they still use antibiotics or some type of hormones, just not as many.
Not with organic. Organic is a very tight label. That's a meaning. Natural is just complete garbage. So natural is the one you're probably thinking of. That just means there's no plastic in there. So. Yeah. Yes, and then, yeah.
Thank you. Has there been any significant legislation from the past that has tried to address these issues? And if you were to propose new legislation today to address this issue, what would be the key proposals you would make?
Right. So Louise Slaughter is our only microbiologist in Congress, and she has repeatedly put forth legislation to reduce antibiotic use in animal production to only therapeutic use, and bring all antibiotic use under control of a veterinarian. And that has been received almost as if you were proposing to require all male children in school wear skirts. It's somehow just super radical that you would only allow antibiotics to be used for sick animals. So Louise Slaughter's bill, it never gets voted on. But it's a great bill, PAMTA, Preservation of Antibiotics for Medical Treatment Act. And I can't remember the name of the Senate side bill, but I think with more and more companies coming on board with saying, like big companies like McDonald's and Tyson and big producers saying, hey, we're going to get rid of non-therapeutic antibiotics, at least in poultry, it makes it a safer environment for Congress to kind of step in, and so we're hoping that a bill like that might gain momentum someday. And God bless Louise Slaughter. What's that?
What's the name of it? One more time,
PAMTA, Preservation of Antibiotics for Medical Treatment Act. And I really like that. There's also legislation, STAR legislation. I don't know if that one's still getting introduced, but that was something that Stuart Levy was a real champion of, which is just real monitoring of antibiotics and antibiotic resistance. And that's one of the things that we need. We need data. I mean, it's ridiculous that antibiotic use can be considered a trade secret. It is a shared resource that needs to be protected by all of us, and it's just a shame. Yeah. Thank you. And FDA has done some things, but not enough.
I heard that the bacteria in pork is very hard to kill. Even if you cook the pork well done, the bacteria is still not killed. I mean, that is, I heard from a Muslim friend. And she said, that's why we don't eat pork. Because it's still the same, and I heard it from somebody who is doing research in NIH, so I don't know whether that is true. To me, bacteria is bacteria, and you kill them at the same temperature wherever they are,
Yeah, there can be a little bit of variation in thermostability, but I'm with you. Most of it is very killable with cooking, and a well done pork chop is not a risk factor. It's handling that raw pork chop that you bring into your house. And there used to be something called trichinosis, and there still is trichinosis that you can get from undercooked pork, but in the United States it's largely absent from our pork production, and that's not even a bacterium, that's a parasite. So we've seen there are some strains of E. coli, for instance, that are a little more heat stable, but nothing that could put up with a good grilling. Yeah. Do I need to shut up or yeah? Play.
I've kind of a question regarding like hand washing and antibacterial soap. Wasn't that recently sort of banned or kind of scaling back the use of it?
Right.
How does that kind of affect or relate to this, or is it like a separate?
Yeah, so there's something called triclosan and triclocarban that they put into soaps. This is the antimicrobials that they put into soaps. And, it never really, it was never, triclosan soaps were never really any better than regular soap. I mean, it's the physical action and the little bubbles that actually take the bacteria off your hands and flush them down the sink, right? But the problem is that those drugs can, in the environment, select for drug-resistant bacteria, but they can also produce these stable toxic compounds that look like dioxin in the environment. And so it was never a good idea. It was just always a sales gimmick. And so I understand that they're trying to ban that now, and it couldn't be sooner. Yeah.
Okay, please join me in thanking Dr.
Oh,
Price.
thank you. Thank you.
This has been a presentation of the Library of Congress. Visit us at loc.gov.